Yes.
Right, so I think we can start now. So, well, welcome everybody. This is my pleasure to introduce Mingming Wu as our last speaker in this program. Tom Powers was our first speaker. He's in the room, so the loop has looped. And it's our pleasure to host you today. So Mingming is a professor in biological and environmental engineering at Cornell University. And what can I say? You are originally from China. You did your PhD at Ohio State, which I've learned this morning.
Yes.
She's well known for a lot of work using experiments to study problems in biology and medicine. For example, well, she'll tell you about cell movement, but she's done a lot of interesting work about cell movement. Also, a lot of important work on cancer. She's a fellow of the APS, and she's here until you're leaving Tuesday. So she's sitting at the Ayana on the first floor. So welcome and thank you.
Thanks, Eric. Thanks for involving me here in this great workshop. I guess it's a program. Yeah, it has been really very informative and a lot of conversation. Met some old friends and made new friends. So that was really wonderful. So I'm going to talk about how physical environment influences cell migration. So in the spirit of Darcy Thompson, I would like to show this movie first. We all know that we started with one cell and then became 2 and 4 and 8, and somehow our cells can always find the right place and form the right organ. So this is the first 18 hours of fruit fly development. And since we were interested in how heart works, so we used this fruit fly where only the cardiac cells are green. So we can see that in the first, this is a 3-hour movie, and the cells actually somehow knows to go to the right place and to form a tuber heart. And so in a different setting, immune cells. So in this case, what you see is neutrophils. So these are our body's defense. So if we get a cut, neutrophils could come in, kill the pathogens. In this case, he is actually chasing a bacterium. And so he could persistently follow the bacterium until it engulfs it. So this is quite amazing things that the cells at single cell level can do. And so here is another example where cell migration is important. And so these are endothelial cells, which is the cells that form our vasculature. And if we place them on a 2-dimensional surface, it forms automatically into this tube shape. So we don't really have to do anything. These cells just automatically do that. And so our question is, why do they do that? Is there any underlying principles that governs it that we can understand, we can use to predict where cells are going to go and what is the function of these cell migration. So before I start, I show a picture of a satellite image of the Earth at night, and you can see that the light is not uniform because people also migrate. And they go to different places for different reasons, and typically it could be temperature or proximity to water and landscape. And although these days politics also play a role. So if you look at the last one, it says Trump makes me want to be Canadian. So that's so Cornell is very close to the border of U.S. and Canada. And the day that Trump won election, the border had a jam. So, there are many factors that can influence migration. So in my lab, I was trained as a physicist, and so we naturally go into a simpler system. So we look at the single cell migration, and we started with the chemotaxis, how chemical gradients influence cells. However, as we go along, we find that higher organism, which means us, mammalian cells, in fact, are quite sensitive to physical environment in contrast to chemical environment. So I'm going to talk about two examples that we have worked on in the lab. One is on how sperm cells decide where to go, and the other is on how breast tumor cells make decisions in a specific environment. So this work, almost every single data I'm showing here is taken by Qi Guangdeng, who was a postdoc in my lab, and he has moved on now a faculty member in North Carolina. And so this is in collaboration with biologist Susan Suarez and her postdoc Flo and theorist Don Koch at Cornell. So this is an image of bovine sperm, and so most of the different species have more or less similar structure. So it has a head, and then a neck, which is a little thicker than the tail, and this is about 1 micron width here, and down to 20 nanometer here. And so the head is about 5 micron wide, 10 micron length, and very thin, 1 micron thickness. So the actuation is in fact along the flagellum. So if we look at the cross section of the flagellum, it has 10 of these microtubule doublet, and each of them is powered by dynin, which is the showing on the red little thing here. So they can contract and relax, and so that contraction and relaxation will allow the sperm flagella to bend and swim around. So if when we put them in the lab, so this is in regular media, and this is in viscoelastic fluid. And so you will see that they swim by wiggling their tail. And they can actually swim pretty fast. In the regular fluid, Newtonian fluid, it's about 130 micron per second. And in the viscoelastic fluid, which is more in vivo-like, it's 50 micron per second. So the question is, what is the driving force? How does the sperm decide where to go? And so a little bit of history of the problem. So when Susan, my biology collaborator, came to my lab, she came because of this device that was developed in my lab. And so we were using this device to generate a chemical gradient. And the way that we generate chemical gradient is by micropattern 3 parallel channel in an agarose gel. And so when we flow attractant and buffer in the side channel by diffusion, you establish a gradient in the middle. And we were reasonably successful looking at the bacterial chemotaxis, immune cell chemotaxis, and tumor cell chemotaxis. So Susan heard one of my talk, and she came to say, well, Mingming, there are a lot of hypotheses to say that the sperm is chemotactic towards the egg. So she wanted to test whether the stuff that the egg secreted is attractant to the sperm. So that was basically the starting point of our work together. And so she had put a graduate student for 4 years. And so these are the experiments. So first, the graduate student Haixing put mouse sperm into the cell channel with the gradient. And however, after 2-3 years, she does not see the chemotactic behavior. So the cells basically do random motion, and then she needs to graduate. Everyone was frustrated, and the student was frustrated, and they said negative result is terrible. And basically, it was not very good collaboration. But then she was very, she's a good student, and she thought about it. She said, well, let's do a control. So when the spring comes, she ordered some sea urchin sperm. And so sea urchin sperm experiment can only actually do 2 times in a year. So spring is one of them. And so she used the sea urchin sperm and did the same experiment, and clearly the chemotactic behavior is evident, because in the absence of chemoattractant, the cells just spinning around, circling around, while in the presence of chemoattractant, it breaks away the loop. And so we knew that there is nothing wrong with the device or experiments. In the end, it is likely that the mammalian sperm just do not chemotactic. And there might be a reason for that, because in a mammalian cell, in the mammalian reproductive tract, it's a defined route. And these mouse sperm swim pretty fast. So from a biological point of view, there is no need for actually a chemotaxis to be there. While for sea urchins, they mate externally and in the turbulent flow. So they actually need this chemotaxis to function. However, during this work, we find that the sperm cells are very sensitive to the flow we introduce in our device, and it also always look for little tiny imperfection in our device. So we decided that maybe it's the physical environment that are more important for these guys. So Susan and I wrote a proposal to NIH, so we always have to do that, and got funded. And basically, our hypothesis is fluid flow and physical environment is guiding the sperm cell migration. So this is the first experiment we did, in which we put the sperm cells in a microfluidic device, and we introduce a flow. And it is immediately evident that in the presence of the flow, the cells actually swim against the flow. So, so this is in the absence of flow, the cells basically make circles, and so curiously, somehow it always makes right-hand circle. And in the presence of flow, the cells all swim against the flow. And so the student who's doing this experiment is he's a postdoc, Qi Guang. So he was trained as a physicist. So as a physicist, we are always interested in order parameter and the control parameter. So so we can use the average orientation against the flow as our order parameter, and the shear force as our control parameter. And when we did that, it actually gave us this similar to what we call supercritical transition in soft condensed matter. So this is basically a very famous for for people who's doing convection or really convection or Taylor quet. So at the unstable point, that the order parameter always go by scaling law of 1/2 with respect to amplitude. So this is exactly what we found. So we thought, boy, this is an interesting phenomena, and we got to be able to explain it. And so we went around and talked to different people. And so Don Koch is a theorist at Cornell. So, and he and I had worked together before. So I went to Don's office, and a few days later, Don said, Mingming, you should read this paper. And so we read this paper, and then a week later, Don and me and Qi Guang, we had a meeting. And so it turns out we could use the explanation here in this paper from Loga and Bartolo can easily explain our experiment. So basically, what happens is that first of all, we have a pusher swimmer. So because the swimmer is rotating, so, and it's rotating close to a surface. So this basically gives us a torque that makes the sperm go around. So this is like I would call ping pong ball effect. If you spin a ping pong ball, it circles around. So this is basically our first term. So if we write an equation for the angle, the orientation, then the first term comes from this ping pong ball effect, and then the second term is from the Bartolo paper, which is from the asymmetry from the front and the back, and because of the structural asymmetry, and so the drag force is different at the front and the back. So this leads to the second term here. So this is a very simple equation that we can solve, and so this equation predicts there is a critical flow rate at which this transition will happen, and also it predicts the whole second order, the supercritical transition. So so this is exact solution to the equation, and then these are the experimental data that has no fitting parameter. So this is, I have to say, the only biological experiments I have done that had such a great explanation and fit extremely well to the theory. So,
Can I just
so
ask
this was
if both mouse and sea urchin behave the same in a flow? Or very
Yes,
good, or they do?
yes, yes, it would. But the critical value would be different. So the requirement, so this is really the beauty of the paper, from the PRL paper is it only required the cell to be pusher and fall back asymmetry. So that's the two things that is required for the swimmer.
Just one thing, their sizes are the same, or one is 10 times bigger than the other one?
The science.
Yeah, the
Size.
size of
Size.
orchid or mouse.
Oh.
I think mouse probably are like 10 or 20 times bigger than cereal, but I'm not sure.
Size.
Yeah.
So
If
so
the
we
scaling.
worked. So we worked. This particular data is from bovine, from cow. The length is about 100 micron, the flagella, and the mouse is slightly smaller, but I don't remember the size from the top of my head. But the shape is very similar.
But they put the two plots in a non-dimensional length, like for a swimmer. If I compete against a spermatozoid, he'll go faster probably.
Which one compete with which?
Longer or shorter?
Okay, so maybe we can talk afterwards. So we're very excited about this, and then we talked to Susan again, our biologist. She always sort of just put us in the ground, and she goes like, well, is this useful? So what? These guys swim against flow. So what? And so it turns out that there are this passaging that is prevalent for cow industry, and there is also a counterpart for human, and it is basically a passaging that reduces fertility, and both in human and cow industry. It's a different. It has a similar name, but exactly same swimming behavior. So when we looked at the way that this passaging swims, it's very similar to what fluid magnesium would call a polar. So it has it has one flagella that wiggles around in front, and then there is also a wave that sends through the body. And so we thought that it is likely that this passaging does not have the ability to swim against the flow. And so the first thing we did is we look whether whether they have a differential swimming behavior close to the boundary. And so in the trajectory, it is clear that the sperm actually really likes boundary. When they hit the boundary, it follows. While the passaging basically does a random motion, so they don't really get guided by the boundary, which makes sense because we know that the polar do not have this ability of swimming along the boundary. And when we put them in the flow, the passagings get swept away, and while the sperm swim against the flow. And so the other thing that Susan, my biology collaborator, pointed out is that in cervix or in human reproductive system, it is lined at the surface. It's lined with these micro-sized channels. This is 20 micron. And in the SEM image they had for bovine cervix, they often find the sperm got stuck in these channels. So since we saw that they like to go into surface so much, that we built a similar cervix on the chip, where we put grooves on one surface, and then we put sperm cells in between, along with the passaging, and see in the presence of flow how does these two work. So this is the percentage of cell gets swept away, and this is sperm with the groove, and this is sperm on flat, and this is the passaging. So what this basically tells us is the groove and the flow helps the sperm to go against the flow, go to wherever they want to go, and then prevent the passagings from going to the same place. So, so our conclusion is that in fertility, sperm is not the only story. So the reproductive tract, in fact, plays as an important role. And we think that they co-evolved, the two of them co-evolved to make the robust sperm migration system. So that's, there is one more story. So that, again, Susan said, well, that's great, but in the real biology system, the fluid is not Newtonian, and it is viscoelastic. So we, my postdoc actually measured the real mucus rheology, viscoelasticity, and made a solution that is similar to the cow mucus fluid. And so what he found is that in the Newtonian fluid, sperm swim individually. However, in the viscoelastic fluid, so this is a lower viscoelasticity and a higher viscoelasticity, and you can see that they group together. So basically, viscoelasticity encourages the cell to group together and form collective dynamics. And so this is basically a quantitative, quantified result. In the standard medium, percentage of cell cluster is 20, 20, while in viscoelastic it went to 60. So what it tells us is that viscoelasticity enhances collective dynamics of swimming sperm. So, so this part of the story is at this point is still not known. So since there are a lot of theories here, and so it's a challenging question for all of you. Why does viscoelasticity encourages the collective dynamics of swimming sperm?
But you have either viscoelastic or not. You don't have some study where you see how the results depend as a function of fluid properties?
So I have so far only 3. So one is standard medium, viscous medium, and viscoelastic medium. Yeah, we don't have a very gradual thing at this point. So this is the in vivo-like situation. This is just regular medium.
And there are probably some dimensionless parameters that govern this.
What is the difference between standard and viscous?
This is just like water. This is a higher viscosity, the same viscosity as the in vivo fluid, but without the elastic part. So this is the in vivo-like.
Did you cool it, or is this all at 37 degrees?
All these experiments are done at room temperature.
Right?
Yeah, they're very sturdy.
Because that's another way to start teasing it apart, is change the temperature.
It's a similar set of experiments to the ones that David Smith and his group do in Birmingham. You've probably seen them. Mainly, it's a lab which concentrates on male human sperm motility assays, and they've looked at a number of similar things. You've probably seen them somewhere.
What's the last name?
Smith?
Smith, yes.
Dave
Yeah,
Smith.
yeah.
And he works with.
That's easy to find.
Yeah.
There's a
David
medic.
Smith.
He's a medic whose last name is Kirkman Brown. So
Yes,
that's not
yes,
so
they published this.
Yeah.
Yes. Okay, so so this is basically half of my story. If you have any questions, right now is the right time.
Now we're at the end. For your biological fluid, it's obviously it's viscoelastic. How shear thinning it is? Is it strongly shear thinning or not?
So I have a plot. The problem is if I go to the plot,
You can maybe at the end.
Yeah. So yeah, it's
But is it or is it not?
it is shear thinning, very shear thinning. Yeah. So we measured both. So it's in our PNA supplementary. We measured both our fluid and the real fluid from the cow.
Question. Do they all have the same or similar Struble number? Like 0.3, the most efficient, or say what's the wiggling frequency compared with the speed and the size?
I've not measured.
Or,
I think the.
If
So
they're
why is Struble
efficient-
number important?
You can derive, like say, 0.3 is more or less the most efficient sometimes.
Efficiency.
But it depends, of
Okay.
course, on the shape, on the size, on the 2-3 dimensionality.
Mechanical efficiency is swimming in an open chamber is maybe not the most pressing pressure, survival pressure on sperm.
I know, but who knows?
Speed.
Also, I think
Or,
0.3 is the
long-distance
happiness number.
endurance.
It's not-
Yeah, these are very low right now. so-
I think with PAV, you would calculate it very
Yeah.
easily from the
Yes,
sequence.
so
If
that's
you wanna
one
let
of
her
the
talk about the
interesting
second half.
questions. What are they optimizing? Is it swimming efficiency, energy, and so these are not. Eric knows. I
don't know.
you know, we don't know yet. Okay, so I'm going to go and talk about the second part of this talk, which is on tumor cell migration. So this is, again, same kind of story. We started with chemotaxis, and then we find that, in fact, it's fluid flow and it's physical environment, gel stiffness, that are much more important for these guys to migrate and grow. So, after a few years, all my projects now are physical force regulation. So this is the, most of the data, again, is taken by Matt Hall, who was a graduate student in my lab. He's a postdoc in Michigan now. And heavily collaborated with the theoretical group. So Xingzhen is a graduate student with both me and Herbert at Cornell, and Farid and Asham are postdoc with Vivek Shenoy at UPenn. So the story also starts with one cell. So the cell basically is supported by this three-dimensional structure. And traditionally, people always plate cells on a 2D surface. And recently, we all realized that what we learned from this 2D surface cell culture actually do not always apply to the in vivo situation. So to give an idea of how cells migrate in 3D, the movie is not playing. It's a very nice movie. So basically, the kids climb the rope and go up. So that is basically how the cells migrate. They use these E-coherent. This is the fibronectin, and they cling onto the rope, and then they pull themselves up. And so the cell typically keeps a balance with the surrounding. And if this balance got broken, then problem arrives. So in cancer, this looking at the extracellular matrix architecture is one of the important diagnoses. And so most cancer types are lethal only when the tumor cells metastasis, which means that they break away from their primary tumor and somehow manage to migrate through the extracellular matrix, squeeze into the vasculature, and take a highway and extravate and establish a second tumor. So the question is, how can they actually do that? In fact, it's not all clear that this is an easy thing for them to do. And so the question we have is, what is the governing principle for them to migrate? So in some sense, in physics, we have F equal ma, which means if you have a gravitational field, you have a mass, you have a principle F equal ma, so you can predict where it goes. So here we ask the same question, that if we know the tumor microenvironment, can we understand the basic principle so we can predict where these cells are going to go? So the first thing clearly is to measure how much force the cells is going to exert onto the matrix. And so there is a history of traction force microscopy development, and from 2D to 3D. And so when we started to look at this problem, we find that the limitation of the current technology is these traction force microscopy is either on 2D surface or in synthetic gel, which is linear elastic. And then we knew that biological gel, like our tissue, they are not linear, they are nonlinear, and they are also fibrous. They have all these networks. So we started to look at it. The first thing we do is to make these gel, change their property, and so that we can test how does the cell behave in these different gel. So one trick we use is to cross-link them, use sugar. So this is in fact very close to the diabetic patients when they have higher sugar level. They tend to cross-link the collagen, which make the tissue have different mechanics as the normal tissue. And the other way we do is to change the temperature. So by doing these tricks, we can get a different gel. And so one of the things we do is, since we are interested in breast cancer, so we made different gel that covers the modulus of normal breast tissue and breast tumor tissue. So the experiment basically goes like this. We put, this is a breast tumor cell, into the collagen, and then we cross-link the beads, this is green fluorescent beads, onto the collagen. So when the cells migrate through, the bead make a displacement, which is basically the deformation of the gel. And so from deformation of the gel and the collagen property that we know, we can back out the force. So that's basically the basic idea behind the traction force.
It's a 3D network, isn't it?
Yes, yes, it is. So so we spent actually a number of years to figure out how to track these beads, because they are very dense, and but we used a lot of fluid mechanics tracking algorithm to do this. And then when we fit to the theoretical model, we find that the importance is to take into consideration of the fiber network. So this from here to here is another a few years. This whole thing is 5-6 years of work. So the problem was that in the beginning we thought, once we take into the nonlinearity of the gel, the problem would be solved. But when we fit the nonlinear model to our data, which is the black here, which is called inside with the blue here, it just didn't fit. So So the breakthrough is really when we take into consideration of the fiber, because when the cells drag on the fiber align, which introduce an isotropy into the system. So that was basically the break point where we actually was able to explain our data. So this is the, we call it, so this is from Vivek Shenoy's group at UPenn. So they developed a discrete fiber network model, which is taking into consideration of the fact that if you put this string onto this network, the fiber actually align. And so when you take that into consideration, the material model can fit to our measurement here. And so use this model, we were able to measure the traction force that the cell exerts onto the collagen. And so the first thing that we noticed is that the cells don't just exert force. They poke onto the matrix, they tug on it until it stiffens. So basically all these are, every little dot is a cell. And so you find that the cells exert force only in this gel stiffening region. So, which means that this nonlinearity is really critical for cell-ECM interaction.
Sorry, these are numerical results?
This is entirely experimental. So this is this.
You have
This
some
is-
lines.
Right. So these lines are the rheology measurement of the gel that we measure, and this is the string. So this is just the string of the cell that exerts onto the matrix. So it's
You don't have a model for this.
no. This is entirely experimental. So so this is basically the point is that the cells exert enough strain to stiffen the gel. So because so this is so this is the same as the rheology data here. So basically, if the gel is linear, it would have been a flat line. And it has started to, so it stiffens around the area when it shoots up. So the point is the cells like tuck on the tuck on the network. It does not do just a little bit. It tucks until the network starts stiffening. So that's something that we think is important for the cells to actually function, because so we have a lot of linear elastic gel that's synthetic, and the cells don't do well. They either die or they don't migrate. They don't do well, and part of the reason we think is because it doesn't have this stiffening property. So this basically says, the nonlinearity is really critical for the cell-ECM interaction. And so this, if we look at the actual fiber, so this is again a measurement. This is the cell. And you can see that the collagen fiber really gets aligned by the cell. So the previous one, you can see that the mesh is quite homogeneous.
Which is the front of the cell? Which is the front end of the cell?
Oh, I don't know. But I would think
Yeah, the
normally
front
It's pulling there.
yeah.
But it's as it moves,
I think
we
okay,
can
yes,
try the
yes,
front.
yes,
Yeah.
sure.
So this is the string cell, the string that the cell exerts onto the matrix, and this is a different matrix. Matrix are different stiffness, and this is the onset for string stiffening. So basically, again, so this is a redundant plot here, and basically again that the cells always exert enough strain to stiffen the gel. So that is something that we think is important for this interaction. And then the other thing is the stiffen. So if we increase the stiffness of the gel, the cells exert more force. So in a way, it's a reciprocal interaction. The cells make the gel stiffer, and stiffer gel makes them exert more force. And so we haven't really done this final experiment yet, but we think this could be the result of sudden tissue stiffening in tumor, also in fibrosis. Because in both cases, the tissue could be just like this, and then suddenly it becomes very stiff. So, and also the cell body also stiffens when we put the stiff gel into the system. So the cells definitely are sensing the environment. So, so as you know, Eric and I talked a few days ago about the sperm. They might also sense the environment. If it's a viscoelastic fluid, they do different things than if it's just regular media. So this is basically in tumor cell case, it's definitely the case that if you have a different physical environment, they do different things. So another interesting thing we find out is that if we change the nonlinearity, the stiffening property of the gel, we find that with more nonlinear gel, the cells broadcast its influence further. So this basically is the displacement around the cell. So if we have a very nonlinear gel, the influence will go much further than if it's a linear gel. So which lead to the question of whether this type of gel, the nonlinear gel, would enhance cell-cell interaction. So which is the case in this movie that in the case of, so this is the nonlinear gel, you can see that the cells start to cluster, while in the nearly linear gel, the cells basically do random motion. So, so basically what we are interested in right now is that, so this is some new data that we just got. So the red and the green are two different cell types. The green is malignant breast tumor cell, and the red is normal cell. And in this case, we changed the adhesion, and we also have data where we change the matrix nonlinearity. So the question is, we have one cell, and the one cell interact with the other cell, and what is the necessary condition for these cells to decide to invade or remain a spheroid? So that is basically something that we are constantly thinking about, which we also don't have a theory about. So it would be really fun if someone's interested in working with us on this.
Yeah, are you changing the nonlinearity by increasing the amount of carbohydrate? Is that what you're
Yes, sugar, basically.
And that doesn't change just the behavior of the cells alone?
It does. It does because all dependent on the collagen concentration. At some collagen concentration it does, so it does change it because it exerts more force when the collagen has sugar crossing the flow. So basically, in this second story, what I want to emphasize is, in fact, nature makes these matrix that is nonlinear, and this nonlinearity enhances cell-cell interaction, which leads to the collective dynamics. So I was looking at Yves Couder's talk, since I was not here in the earlier talk, and he was talking about biological convergence. And so in this case, I want to point out there is the similarity from the two examples is that natively derived materials somehow always promote cell-cell communication. So one question is whether it's designed this way or it just happened by chance. With that, I would like to thank all the people who have worked on these projects. So Matt and Qigong are the two main people who worked on this project, and many collaborators and funding agencies. So most of these work are funded by NIH at this point. Well, and thank you for staying for the last talk of the program.
There were really a lot of questions, but maybe we have time for a couple more. Yes, Roberto?
So, I don't know if I understood completely the last part. So the fact that the matrix is stiffening is what promotes the migration of the cells? Is that the main idea? Or?
Not, no, it's promote more force generation, which, yes, so which could lead to more migration, but that's not, that's a good question to work on.
I mean, if I were a cell, I would probably want to move where it's easier, right? So you would move to a
Maybe that's why you're not a cell.
No.
Maybe that's why you're not a cell.
Well,
It's not the case.
you know what I'm saying, right?
They like to go to hard places. Yeah, it's amazing. I have some movies where we suspend. So, and also it's more malignant cells does that. So we have
They're
super,
really bad. They go.
yeah,
they're
really
stiff.
bad cells. Yeah, so a very like the most malignant breast tumor cell we worked with, we can't even hold them in the center because they go like this because they like the surface because the surface is hard. Yeah. So, but there are controlled work where people have a gradient of stiffness and cell like the stiff area. Yeah.
Interesting.
There are two. Let's first the back.
I was wondering, the final video, is your open question like how, when a cell modifies the environment around it, how do those cells respond to that? And then is that essentially an interaction through the matrix? So you're asking, is there cell-cell communication independent of the matrix, or?
Both.
Okay.
So the question is, in the spirit of form growth, I think this program is really fantastic. The form growth and self-organization. So now, I would put the environment clearly. So you have a cell and you grow into many cells. For tumor cell case, typically it starts with one cell, and then they grow into 2 and 3 and 4, and sometimes they cluster together, sometimes they don't. So and this is a function of the matrix, a function of their own property, which is growth, adhesion, and their own stiffness and their force generation. So the question is, we endow the cell this property, can we predict whether it forms a spheroid or it would invade? So we we played with many different cell lines and also matrix, and we find in certain matrix they stay put, they don't do anything. But in some other, especially nonlinearity, definitely promotes invading.
Leontine had a question.
Looking at by the way, this was a fascinating. Looking at the video for the metastasis moving through the network, it seemed to me that say at the back of the cell it was pulling, and then at some point it cuts off the adhesion, and then
Yeah, so it's a very dynamic. So in the very dense matrix, the cells actually can dig a hole using MMP, the protein, something to digest and dig a hole and walk very fast. And in the lower concentration case, they literally do this. They hold the rope and so your question is, when they do that, there is
the back, but
asymmetry.
it's not the back, because it seemed to me that it leaves the. If you look at the video,
This one?
yeah. Can you see that
Snaps, right?
as soon as it snapped, it moves quite a bit forward.
Yes.
So I was thinking if that's also something to take into account.
So this is actually our, so we have a new model now, which we're still working on it, is so we can actually look at what's inside the cell. And inside the cell, there's another network, which is the actin filament. So there is an actin-myosin network that we are modeling now, well, Vivek is modeling now. And then they are actually very much connected to the outside network. So if the outside network is aligned, the inside is aligned as well. And so there is some energy balance between inside cell and outside cell. So at the moment, all the stuff I talked about has not taken into that consideration. But yeah, yes, it's whatever is going on inside the cell in the end determines where the force comes from.
Okay, I think because it's already 5 past, maybe I propose we stop. We thank Mingming. Thank you all for coming to all the seminars, I guess, this term.